So, Boyett. Thank you very much. Could we switch these lights off, please, just for this one talk? Thank you very much. So, like others, we're working towards the development of a mathematical model of the entire heart. Now, Helena's laboratory and my own, we focus on the cardiac conduction system. We're building anatomical models and action potential models. And first, these anatomical models. So in 2005, we published a model of the pacemaker, the sinoatrial node of the rabbit heart. Here the model is superimposed on a diagram of the heart. Here the model is shown as a movie. It's superimposed on a simple wire mesh model of the right atrium. Now we're using these models to compute action potential conduction through the tissue. And so here we're using the very simple cellular automaton model to calculate the activation sequence of this preparation. And the calculated activation sequence is very similar to that seen in experiments. Just last year, we published a model of the atrioventricular node of the rabbit heart. Here it is on a diagram of the heart. A window has been cut in the right atrium here so that you can see the atrioventricular node located at the base of the right atrium. And remember that the atrioventricular node is the only conduction pathway for the action potential from the atria to the ventricles. So here is the model shown as a movie. Again, we're using this model to compute action potential conduction. Here we have used biophysically detailed action potential models. And you're watching a simulation here of a common arrhythmia that Alan mentioned, atrioventricular nodal reentrant tachycardia. Now, these two models were both based on histology and immunolabeling of marker proteins. Now, a more recent model instead is based on high-resolution magnetic resonance imaging, and this was carried out by Jürgen Schneider at the University of Oxford. And we have used this to construct a model, an anatomical model of the entire right atrium of the rabbit heart. So this is an example of one of these images. This is the right atrium, the right ventricle's down here somewhere, and here are leaflets of the tricuspid valve separating the two chambers. Now, in the rear wall of the right atrium, you've got a thick bundle of atrial muscle. It's called the crista terminalis, and it's a very useful landmark in our work. And perhaps you can see that the tissue next to it is translucent. And this is the nodal tissue making up the pacemaker, the sinoatrial node. Now, there is another patch of translucent tissue here on top of the interventricular septum. And this is part of the atrioventricular node. So this demonstrates that we were able to identify the nodal tissues in these images. So from these images, we constructed an anatomical model of the entire right atrium of the rabbit heart, and you're looking at 4 different views of this model here. At the top left, you see the exterior of this model. Now, this model has been segmented, and at the present time, we have segmented 18 different structures. So the superior and the inferior vena cava are shown in blue, and running between the two in red here is the sinoatrial node. This is another exterior view of the right atrium. This time the atrial muscle is translucent so that you can see the atrioventricular node, here it is, within this structure. At the top right is an interior view. Now in purple is the thick bundle of atrial muscle, the crista terminalis, and running alongside it in red is the sinoatrial node. You can see various landmarks, the fossa ovalis, the inferior vena cava, the coronary sinus, and in yellow here you see leaflets of the tricuspid valve leading to the right ventricle down below. Now, running from here to here is another tract of nodal tissue. This is the nodal tissue forming the atrioventricular node. Now, down below is another interior view focusing on this atrioventricular node. Now, part of the nodal tissue, it's shown in this green color here, is enclosed in fibrous tissue, and we call this the penetrating bundle. And this represents the tract of nodal tissue as it passes from the right atrium into the ventricles through a fibrous layer. Now, some of the nodal tissue projects into the right atrium. It's shown in red here, and it's called the inferior nodal extension. And Igor Efimov has shown that this is the slow pathway into this penetrating bundle. Now, once again, we're using the model to compute action potential conduction. And here we've used the Fitzhugh-Nagumo equations. And this is a simulation of atrial fibrillation, which we've triggered using an S1, S2 protocol. On the left, you see an exterior view of the right atrium, and you see high frequency activity. On the right is an interior view focusing on the node. Now notice that although you've got high frequency activity within the atrial muscle here, the frequency of action potential is being conducted along the node to the to the ventricles is very much lower. And this is a well-known protective phenomenon, and is the result of the relatively long refractory period of the atrioventricular node here. So this year we started to construct a model of the His-Purkinje network in the rabbit heart, and this work is being led by Helena Dobrinsky. And for this work, she cut off the atria, she cut open the ventricular free walls to gain access to the ventricles. She then immunolabeled a marker protein, which is only expressed by the conduction tissue. And this is a photograph of the His-Purkinje network within the left ventricle. I hope you can see it, and it's very elaborate and very complex. And perhaps the His-Purkinje network on the right side of the heart is even more complex, as you can see here. Now, this is a high magnification view of the left bundle branch. Now, when we started this work, I was expecting the left bundle branch and the right to be a large diameter bundle of specialized fibers, but this is not the case. The left bundle branch has the structure like a ribbon cable, and it consists of a large series of fibers running in parallel, and these fibers are only 1 myocyte in thickness. Now, this is a high magnification view of the His-Purkinje tissue in the left ventricle of another heart, and I think this is really very beautiful. And it shows, for example, that you have Purkinje tissue here and here, which is pale staining, and this is Purkinje tissue on the surface of the ventricles. But other Purkinje tissue takes up the stain more darkly, and these are free-running Purkinje fibers. So you've got two populations of Purkinje tissue. And we've looked at this in the left ventricle of one of the hearts. In blue here, you see His-Purkinje tissue, which is attached to the surface of the ventricle. And this accounts for about 70% of the total tissue. And in red here are the free-running Purkinje fibers, accounting for about 30% of the tissue. We were able to distinguish at least 2 layers of Purkinje fibers, these free-running Purkinje fibers, and we got a few here, shown in green, in the top layer. Here we have merged the 3 layers to form the entire system once more. Well, here are the left and right Purkinje networks, and already we're beginning to use these to compute action potential conduction. Here we've used the cellular automaton model, and you can see slow conduction through the atrioventricular node, followed by very rapid conduction through the His-Purkinje networks. Not only are we producing anatomical models, we're also generating action potential models. In 2000, we published models of the action potentials in the rabbit sinoatrial node, and this year we're publishing models for the action potential in the 3 major cell types at the atrioventricular node: the transitional AN cells, the true nodal cells or N cells, and these transitional NH cells. And I want to show you an example of the use of this new family of action potential models. So conduction through the atrioventricular node is slow, and it's under the control of the vagal nerves, which of course release acetylcholine. And acetylcholine is controlling conduction velocity, and we're investigating how. Well, this is a simulation of the effects of acetylcholine on the action potential in our 3 cell types here, and also we've looked at the effect of acetylcholine on an ordinary atrial action potential. We're simulating acetylcholine by activating the acetylcholine activated potassium current, by decreasing the L-type calcium current, and by shifting the IF activation curve. You see in most cell types there's a marked shortening of the action potential. In the N cell and the AN cell, you can see that at the highest acetylcholine concentration, acetylcholine abolishes the action potential. But remember, we're really interested in the effect of acetylcholine on conduction velocity. So we have constructed a one-dimensional model of the conduction pathway through the atrioventricular node. It has slow and fast pathways, and it incorporates the 3 cell types. Now, we stimulate the atrial end of this string, and we're recording action potentials at these 4 sites here to observe what happens to conduction velocity. And you see here action potentials at these 4 recording sites under control conditions and with acetylcholine. The action potential is triggered in the atrial muscle. The action potential propagates to point 2, which is in the fast pathway at the end of this string of AN cells, in about the same time without and with acetylcholine. But conduction along this string of N cells is greatly slowed by acetylcholine, and so the action potential reaches point 3 at a longer time in the presence of acetylcholine. And it's because of this that the action potential reaches the end of the string here, point 4, at a longer time in the presence of acetylcholine. And this shows that this conduction time increases as we increase the acetylcholine concentration. Well, this, of course, is a highly simplified model, and so most recently we have started to use our new family of action potential models together with our 3-dimensional model of the atrioventricular node. And you're going to see, when I rerun the movie, conduction first through this node under control conditions. And the action potential is propagating through the node normally on its way to the ventricles. Now, the same thing in the presence of acetylcholine. And what you're going to see is that the action potential is blocked at this point here. And it's well known that excessive vagal stimulation can result in heart block, in other words, in a block of conduction through the atrioventricular node. And that is as far as we've got. Thank you.
Have you done several hearts in doing the Purkinje system, getting the Purkinje system? Because I understand there is a very large variability between hearts. And would there be any rule that we would be able to extract to not be needing to reconstruct every single heart?
Well, remember this work, the PI behind this work is Helena Dobrinsky, and she's not here today, but she has given me permission to show you these data. She's looked at 3 hearts, or we've looked at 3 hearts so far, and this work is ongoing, so I think it's just impossible for me to draw any conclusions at this moment, but this is something we're going to be working on, and we've certainly got a 4th heart lined up, so we're going to be doing more of this.
And would you be able to figure out what is the penetration of these Purkinje? Because different species have different penetration.
When we first started doing this work, see, the immunolabeling I've shown you is on the entire heart. But when we first started this, we started off with tissue sections, and we could never find any Purkinje tissue. I was looking for these great big left and right bundle branches, but they don't exist. And transmural Purkinje tissue doesn't exist, as far as we can see, in the rabbit heart.
In the rabbit, yes, exactly.
I know that
the pig, yes.
Maybe. So, but in the rabbit, there's no transmural Purkinje tissue at all, as far as we can see.
Okay. Yeah, just to follow on that one. When we have done it in dogs, I was surprised to see in your case that most of the Purkinjes are floating Purkinjes more than they're floating. You have more percentage of floating Purkinjes.
It was 70% in that one heart, in the left ventricle. 70% was attached, and 30% was floating, if you like.
30%. Okay, yeah. So in the dog we find like a lot, like also
yes.
70% is just on the tissue.
Yeah,
and
yeah.
It doesn't go also inside like the pig. It's only superficial.
Yes, yes. Well, exactly the same then. Yes.
Okay, we should move on to part 2.